This video shows our methods for high-throughput siRNA screening in a corneal epithelial cell model of toxicant injury. This work is important because high-throughput siRNA screening is a cost-efficient and time-efficient method to elucidate pathways and regulators of the cellular response to toxicants. This work may help further our understanding of how the cornea responds to toxicant insults. Prepare a suspension of cornea epithelial cells in pre-warmed medium containing 0.5 micrograms per milliliter hydrocortisone in a disposable medium bottle. Swirl the bottle to evenly suspend the cells and pour into a reservoir on the orbital shaker nest of an automated liquid handler. Keep the cell suspension on a 37-degree warming plate during the cell plating process. Use an automated liquid handler to add cells to plates at a density of 1,000 cells per well in 70 microliters of medium per well of a 96-well plate. Seed a minimum of 14 96-well plates with cells for every siRNA library plate to be studied. Run the orbital shaker at 100 cycles per minute constantly during the seeding process, and use a 50 microliter per second pipetting speed for all steps. Mix the cell suspension using the automated liquid handler before seeding the first plate. Aspirate 140 microliters of the cell suspension and dispense 70 microliters into each well of 2 cell culture plates. Repeat the mixing and dispensing until all plates have been seeded with cells. Refill the cell suspension reservoir as needed. After the plates have been seeded, remove them from the liquid handler and incubate them for 30 minutes at room temperature, then transfer them to a cell culture incubator. Evaluate the cell density of all plates the following morning using an automated imaging system which is housed in a cell culture incubator. Exclude plates from study which have interior wells that are not between